Hello, welcome to Biology, the scientific study of life. Again, the solvent of life, more about water. A sugar cube placed in a glass of water will dissolve. The glass will then contain a uniform mixture of sugar and water. The concentration of dissolved sugar will be the same everywhere in the mixture. A liquid that is a completely homogeneous mixture of two or more substances is called solution, a solution. The dissolving agent of a solution is the solvent, and the substance that is dissolved in the solute. In this case, water is the solvent and sugar is the solute, and aqueous solution is one in which water is the solvent. The medieval alchemists tried to find a universal solvent, one that would dissolve anything. They learned that nothing works better than water. However, water is not a universal solvent. If it were, it would dissolve any container in which it was stored, including our cells. But water is very a versatile solvent, a quality we can trace to the polarity of the water molecule. Suppose, for example, that a crystal of the ionic compound sodium chloride Na and chloride Na and also NaCl is placed in water. So ions are exposed to the solvent. These ions are and the water molecules have a mutual NaCl is placed in water. At the surface of the crystal, as we see here in this picture, at the surface of the crystal, the sodium and the chloride ions are exposed to the solvent. These ions and the water molecules have a mutual affinity through electrical attraction. The oxygen regions of the water molecules are negatively charged and cling to sodium cations. The hydrogen regions of the water molecules are positively charged and are attracted to chloride anions. As a result, water molecules surround the individual sodium and the chloride ions, separating and shielding them from one another. The sphere of water molecules around each dissolved ions is called a hydration shell. Working inward from the surface of the salty crystal, water eventually dissolves all the ions. The result is a solution of two solutes, sodium cations and chloride anions, homogeneously mixed with water. The solvent other ionic compounds also dissolve in water. Seawater, for instance, contains a great variety of dissolved ions, as do living cells. A compound does not need to be ionic to dissolve in water. Compounds made up of non-ionic polar molecules such as sugars are also water soluble. Such compounds dissolve when water molecules surround each of the solute molecules. Even molecules as large as proteins can dissolve in water if they have ionic and polar regions on their surface. Many different kinds of polar compounds are dissolved along with ions in the water of such biological fluids as blood, the sap of plants, and the liquid within all cells. Water is the solvent of life. Hydrophilic and hydrophobic. Hydrophilic and hydrophobic. Whether ionic or polar, any substance that has any affinity for water is said to be hydrophilic, from the Greek hydro, water, and philios, loving. In some cases, substances can be hydrophilic without actually dissolving. For example, some components in cells are such large molecules or complexes of multiple molecules that they do not dissolve. Instead, they remain suspended in the aqueous liquid of the cell, such as mixture. A mixture is an example of a colloid, a stable suspension of fine particles in a liquid. Another example of a hydrophilic substance that does not dissolve in cotton is cotton. A plant product cotton consists of giant molecules of cellulose, a compound with numerous regions of partial positive and partial negative charges associated with polar bonds. Water adheres to the cellulose fibers. Thus, a cotton towel does a great job of drying the body, yet does not dissolve in the washing machine. Cellulose is also present in the walls of water conducting cells, as in a plant. You read earlier how the addition of water to these hydrophilic walls allows water transport to occur. There are, of course, substances that do not have an affinity for water. Substances that are non-ionic and non-polar actually seem to repel water. These substances are said to be hydrophobic, as it is written here. From the Greek phobos, fearing. An example from the kitchen is vegetable oil, which, as you know, does not mix stabili with water-based substances such as vinegar. The hydrophobic behavior of the oil molecules results from a prevalence of relatively non-polar bonds, in this case bonds between carbon and hydrogen, which share electrons. Almost equally hydrophobic molecules related to oils are major ingredients of cell membrane. Imagine what would happen to a cell if its membrane dissolved. Solute concentration in aqueous solutions. Biological chemistry is wet chemistry. Most of the chemical reactions in organisms involve a solute dissolved in water. To understand chemical reaction, we need to know how many atoms and molecules are involved. Thus, it is important to learn how to calculate the concentration of solutes in an aqueous solution, the number of solutes molecules in a volume of solution. When carrying out experiments, we use mass to calculate the number of molecules. We know the mass of each atom in a given molecule, so we can calculate its molecular mass, which is simply the sum of the masses of all the atoms in a molecule. As an example, let's calculate the molecular mass of table sugar sucrose, which has the molecular formula C12H22O11. In round numbers of Daltons, the mass of a carbon atom is 12, the mass of a hydrogen atom is 1, and the mass of an oxygen atom is 16. Thus, sucrose has a molecular mass of 342 Daltons. Of course, weighing out small numbers of molecules is not practical. For this reason, we usually measure substances in unit called moles. Just a dozen always means 12 objects. A mole represents an exact number of objects, as it is written here. 6.02 by 10 23, which is called Pythagoras number because of the way in which a Pythagoras number and the unit Dalton were originally defined. They are 620.02 by 1023 Daltons in 1 gram. This is significant because once we determine the molecular mass of a molecule such as sucrose, we can use the same number 342 but with the unit gram to represent the mass of 6.02 by 1023 molecules of sucrose, or 1 mole of sucrose. This is something called the molar mass. To obtain 1 mole of sucrose in the lab, therefore, we weigh out 342 grams. The practical advantage of measuring a quantity of chemicals in moles is that a mole of one substance has exactly the same number of molecules as a mole of any other substance. If the molecular mass of substance A is 342 Daltons and that of substance B is 10 Daltons, then 342 gram of A will have the same number of molecules as 10 gram of B. A mole of ethyl alcohol C2H6O also contains 6.02 by 1023 molecules, but its mass is only 46 grams because the mass of a molecule of ethyl alcohol is less than that of molecule of sucrose. Measuring in moles makes it convenient for scientists working in the laboratory to combine substances in fixed ratios of molecules. How would we make a liter L of solution consisting of 1 mole of sucrose dissolved in water? We would measure out 342 grams of sucrose and then gradually add water while stirring until the sugar was completely dissolved. We would then add enough water to bring the total volume of solution up to 1 liter. At the point we would have 1 molar solution of sucrose. Molarity, as it's written here, molarity, the number of moles of solute per liter of solution, is the unit of concentration most often used by biologists for aqueous solutions. Thanks for listening.